Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. In today's class I am going to describe the histological model of the kidney. So in this section you can see this is the cross section of the kidney. Kidney is a bean shaped reddish organ located on each side of the abdominal cavity, and it has a medial end which is convex, which has an indentation which is called the renal, where the renal pelvis is located, and this area is called the hilum of the kidney. This outer convex border is covered by the renal capsule. So beneath the renal capsule you can see this dark color area which is the renal cortex, and then you can see the renal pyramids which are present. These are conical shaped structures which are present in the medulla of the kidney, and then the tip of the kidney you can see the minor and the major calyces which join to form the renal pelvis and then the ureter. So in the cortical region, this is the cortical area where most of all of the nephrons are located in different locations, and in the medullary area you can see the collecting ducts, the straight tubules of the kidney. So the whole one pyramid along with its surrounding cortical tissue is called one lobe of the kidney. So lobulated arrangement of the kidney is usually apparent in the fetal kidney, but because of the growth of the kidney in the later stages, so in adult stages it is not possible to see the lobulated kidney. So this is the one lobule, then the two lobules and three lobules. These are the different lobules. So the cortical tissue which is present on sides of the pyramid is called the cortical columns of Burdine. So this one is the cortical column of the Burdine which is present over here, and this is the pyramid, and these are the cortex and the tip of the papilla which is entering into the minor calyces, then major calyces, and then the renal pelvis. So this is the cross section of the kidney. Now we come on to the structure, the cross section of the renal corpuscle, and in this renal corpuscle you can see that this hole is the renal corpuscle. It is formed by the tuft of the capillaries which are covered by the podocytes. So this one is the afferent arteriole coming inside the glomeruli and breaking up into large number of the capillaries, and these capillaries are surrounded by the podocytes. These podocytes are forming the visceral layer of the glomerular area, this Bowman's space, and these podocytes have the food process. So these are the primary food processes of these podocytes, and here you can see these are the secondary and the tertiary food processes of the podocytes which are present over here. Then they join, these blood vessels join to form the efferent arterioles. Here they form the efferent arterioles. This one is the visceral layer of the Bowman space, and this layer is the parietal layer of the Bowman space. So this all area is the Bowman space where the filtrate of the plasma is present over here forming the urine. So this parietal layer is lined by the simple squamous epithelium, and here you can see this is the start of the proximal convoluted tubule where you can see the cuboidal shaped cells which are present over here. Also you can appreciate the juxtaglomerular apparatus over here. So juxtaglomerular apparatus consists of three basically parts: the macula densa, the juxtaglomerular cells, and the extraglomerular mesangial cells. So macula densa, these are the cells of the distal convoluted tubules which come to lie closer to its parent glomeruli, and here the cells, they are taller and they are crowded. They are so close to each other that it gives an appearance that the nuclei override each other, and because of this appearance, their arrangement, they gives a darker color in H&E staining. That is why it is called dense spot or macula densa. So one of the component of the juxtaglomerular apparatus is the macula densa. Second component of the juxtaglomerular apparatus is the Juxtaglomerular cells. These are the modified smooth muscle cells of the tunica media of the efferent arterioles, and sometimes also the efferent arteriole may take part in the formation of the juxtaglomerular apparatus as well. And third component are these cells. These are extraglomerular mesangial cells. So these three components, they are forming the juxtaglomerular apparatus over here. This is the section of the nephron, and in this section, this you can see this is one is the renal corpuscle, in which the tuft of the capillaries are present, which is surrounded by the podocytes, and podocyte forming the visceral layer of the Bowman space. This area is the parietal layer of the Bowman space, and it is lined by the simple squamous epithelium. This leads into the proximal convoluted tubules. So here you can see the convolutions of the tubule. This proximal convoluted tubule then becomes the proximal straight tubule, and then it becomes the loop of the Henle. This is the thin descending segment of the loop of Henle. Then here you can see the hairpin turn of the loop of Henle, thin ascending segment of the loop of Henle, thick ascending segment of the loop of Henle, and then you can see the distal straight tubule and distal convoluted tubule. This distal convoluted tubule here you can see is coming back to the renal corpuscle of its parent origin, and here it takes part in formation of the macula densa. So in this section you can see the different cross sections of the different parts of the nephron. This one is the section from the proximal straight tubule. Here you can see the epithelium is simple cuboidal epithelium. Large cuboidal cells are present. The striated border on luminal side. Also you can see basal striations which are present on the basal area of the cell, and these basal striations are because of the infolding of the plasma membrane on basal side, and in between in these folding the vertically elongated mitochondria are present, and this infolding and vertically elongated mitochondria, they gives a striated appearance in light microscopy. So this is one is the cross section of the proximal convoluted and proximal straight tubule, and this one is the cross section of the distal tubule. So in this distal tubule, in which you can see also the cells are simple cuboidal in nature, just like the cells of the straight proximal tubule, but the difference you can notice between these two cells, that these cells are smaller as compared to these one. The size of the cell is smaller, and another point which you can differentiate between the two sections is that the both having the same caliber, but the number of the nuclei per profile are more than that of the proximal tubule. So in proximal tubule, as the cells are larger in size, so number of nuclei are less as compared to the distal tubule, in which the cells are smaller, so you can see large number of the nuclei over here. This one is the section of the loop of the Henle, in which you can see the simple squamous cells which are lining the loop of the Henle. So this one was the cross section of the cross section and different sections at the different cross sections of the proximal, the distal, and the loop of Henle. Now we move on to the types of the nephrons which are present over here. So in this section, you can see first we classify the nephrons on the basis of location into three. One which is just closer to the cortex is called cortical nephron. The one which is very close to the medulla is called the juxtamedullary nephron, and the one which is in between these two is called intermediate type of the nephron. So these are the three types of the nephron, and their parts. The here is the glomerulus, the renal corpuscle, the proximal convoluted tubule, proximal straight tubule, loop of Henle, distal straight tubule, distal convoluted tubule, and then they all ending into collecting duct. Here you can see the connecting duct as well, and then the collecting duct, and these collecting ducts then form the duct of the Bellini, which at the tip of the papilla enters into the minor calyces. Again, another thing you can notice in this model as well, that the blood supply, which is very important for the kidney, the renal arteries give rise to the interlobar arteries. So these are the interlobar arteries. In these interlobar arteries, then at the junction of the cortex and medulla, give rise to the arcuate arteries, and these arcuate arteries then give an ascending branch which is called interlobular artery, and this interlobular artery runs towards the cortex, towards the capsule, and on its way it gives rise the efferent arteriole to the adjacent nephrons. So in this area you can say this is the efferent arteriole to the nephron, the efferent arteriole coming out of the nephron and its peritubular network. So those nephron which are cortical in origin, their efferent arteriole give rise to the peritubular network in the cortical area, but those nephron which are juxtamedullary in position, their efferent arteriole then they descend sometimes up to the level of the tip of the pyramid, where they take a hairpin turn, and there they form the peritubular capillary network, which is a very important part of the counter current multiplier system. So this one was the interlobar arteries, then the interlobular and the arcuate arteries, the efferent arteriole, the efferent arteriole, and the peritubular capillary network all over here. And this one was the renal corpuscle, the proximal convoluted tubules, the proximal straight one, the loop of the Henle, taking a hairpin turn, then the straight ascending segment, and then this distal convoluted tubule coming back to its parent glomeruli to take part in the formation of the juxtaglomerular apparatus, and then it enters through the connecting tubules into the collecting tubule, and these collecting tubules, when it becomes larger, it is called the duct of the Bellini, which at the tip of the papilla, also called the area cribrosa, opens up into the minor calyces. So this one, the section different types of the nephrons. Here you can also see the branches of the interlobular artery to the adjacent nephrons which are present over here. So these are different type of the nephrons. This one was the cortex, the medulla, this was the blood supply. Here you, in this area, you have studied the nephron and its different histological features in detail. Here you have studied the renal corpuscle along with the juxtaglomerular apparatus, the visceral and the parietal layer, the proximal tubule, the podocytes, and in this section, in the start, we have studied the cross section of the kidney. So this was all about the histological model of the kidney. Inshallah, we will meet next with the next model in the next class. Till then, Allah Hafiz.